Hey biology guys, this is going to be our first video of notes over chapter 11. Some of this should be pretty familiar to you. This is kind of just a little more detail from what we learned when we were talking about DNA in our macromolecules chapter, but we're going to really need to have this stuff fresh in our mind as we go further into this chapter. So DNA, long name, deoxyribonucleic acid. What a mouthful. But the Na is just nucleic acid. Remember, that's a polymer of nucleotides, a type of macromolecule. And then the D is from deoxyribo. Sometimes you'll see it written deoxyribose. Either way. It's a polymer of nucleotides, and it is used to store information. Okay, so I like to think of DNA as kind of the big manual of instructions if you were going to build some large, complicated piece of furniture. Okay, it's the instructions. It stores the information. If there's nobody there to read the information, to put the pieces together, nothing's going to happen. That piece of furniture is not getting built, right? So DNA by itself doesn't really accomplish very much. It's just for storage. Okay? When we want to actually read the information, follow the instructions, we need ribonucleic acid. So again, there's the RNA. That's what is going to be used by organisms to read and follow the instructions that are in the DNA. Okay, so they're a good team. This is storage of the instructions, and this is actually carrying out what the instructions say. And so this is a reminder, nucleic acids are monomer, which remember just means building block, or subunit is the nucleotide. And every nucleotide is made out of three things. It has a phosphate, which is actually a PO4, which is a negatively 3 charged thing. Don't worry too much about that. A sugar and a nitrogenous base. Okay, so here's a RNA nucleotide and a DNA nucleotide. They look very similar, right? So there's the phosphate group, usually drawn with a circle. Again, it's not really a circle, but we usually draw it that way just to kind of simplify things. We have a sugar, that's this pentagon thing. And if you look, the sugars are almost the same. The only difference really is there's an oxygen here, and there's not one here, right? So deoxy would mean without an oxygen, right? So there is actually a bunch of oxygens throughout the sugar molecule here, but it is minus this one oxygen that ribose has. So the deoxyribose name makes sense because it is D and oxygen, without an oxygen. And then there's a base. Okay, and in RNA, you see here's our 4 bases. It could be an A, a C, a G, or a U. The U is bolded because that's the one that's different. And in DNA, it could be an A, a C, a G, or a T. Again, T is bolded because that's the one that could be different. All right, so in DNA, the sugar is deoxyribose. In RNA, it's just ribose. I kind of talked about this on the last page there. In DNA, the nucleotides can be adenine, which we usually just kind of abbreviate these bases with the letter they start. So if you just want to remember the letters for intro biology, I think that's probably okay. Thymine, which is a T, cytosine, a C, and guanine, a G. And RNA, they're almost the same, except there's a uracil instead of thymine. So anywhere that you would think a thymine should go, put a uracil instead. We have this idea called base pairing. that in DNA, certain of these bases, remember these represent the bases, pair up. And they're always the same ones because they actually fit together shape-wise. Their shapes are complementary. And if they tried to match up with another one, they wouldn't fit. Kind of like trying to fit two pieces of a puzzle that don't go together. It wouldn't work very well. So here's our mnemonic, one way to kind of remember which ones go together. So the apple's in the tree, which tells us A pairs up to T. And car's in the garage, which tells us C pairs up with G. And another thing about DNA is it's generally double-stranded while RNA is single-stranded. I stole this picture from Amoeba Sisters. Hopefully they don't mind. So here's our summary of differences. We've got double-stranded for DNA, single-stranded for RNA. We've got the sugar deoxyribose. We've got the sugar ribose. It's generally double-stranded, dental or single-stranded. And then we've got the bases down here. Again, for emphasis, there's the only part that's different. Okay. So the structure of DNA was a major mystery. Okay, so this is in the 50s. There's this great movie about it called Life Story, Race for the Double Helix that I usually show in biology. I might put it online for you to watch if you want to. We'll see if I can make that work or not. But it's a mystery until James Watson and Francis Crick built a model of DNA that finally supported all the data at the time. So they knew nucleic acids existed. They knew proteins existed. They knew lipids existed. But nobody really had known for sure which of those molecules stores the genetic information, the things that cause our traits, the things that pass from parent to offspring to give instructions. And when they kind of figured out the structure of DNA, they figured out how perfect this molecule would be to be the genetic molecule, because if I have one strand, I can recreate the other strand. So it's going to be something that's easily copied, and so it would be really good at sending instructions. Unfortunately for science, ethics, and the rules of right and wrong, they figured this out by using, without her permission really, data figured out in the lab by a woman named Rosalind Franklin, who is famously overlooked and sad because she really should have got a Nobel Prize, but by the time these guys got their Nobel Prize, she had passed away and didn't, and definitely due to the experiments that she did to figure out what DNA was like. So, not a happy story for Rosalind Franklin, but now, for the last 20, 30 years, every time any teacher talks about this, they say, yeah, but she actually had the data. But we do still call it the Watson and Crick model of DNA, so, winners all around, kind of. Okay, so sequence. Okay, when you're ever talking about something's sequence, you're talking about what order does it go in, right? So if I could put kids in sequence by their birthdays, or I could put kids in sequence by their height, but sequence just means a particular order. So the order or sequence of the bases in the middle of DNA is how we store information. And I always think this is super fascinating, and we always have a really nice conversation about this in the classroom, but not this year. Every human's DNA is 99.9% identical. So me and some stranger in China I've never met, not really related to, our DNA is still 99.9% identical. Okay? Or me and somebody who lives in Germany I've never met. At least 99.9% identical. Maybe more because I have a lot of German heritage. But the idea is, this is exactly the same for every single human, which means only 0.1% of DNA accounts for all of the variety of all the humans in the world, which is pretty nuts. Okay, so the 99.9%, why would we need so many of our instructions to be the same if we are really so very different from each other? Okay, so we have different heights, different builds, different levels of coordination, different eye color, there's all kinds of genetic diseases and traits that different people have. So what's that 99.9% that we're all the same have to be about? Okay, so the 99.9 is about how to make cells that work and how to become a human in general. Okay, so if you think about all humans, we are a lot more alike than we are different, right? So we're all made of cells. We all have the same, barring there's a few, obviously the outliers, the people that have severe abnormalities or just, trying to think of the word here, abnormalities or disorders. But for the most part, we've got the same general body plan. We've got a spine, and we have all the same organs, and all of our organs have to have instructions to do all of these important jobs. So if you think about all of the ways that humans are the same as each other, it kind of starts to make sense that our differences are less than 1% of our DNA. So if that 99.9% was changed, the part that makes us all the same, then we might have some vital, very, very important functions that might not work. So that 99.9% is kind of like how to make a functional human. And then the 0.1% is just all those tiny little differences that make us not the same. Okay, so this is kind of an important idea. The less similar two species DNA are, the less closely related they are. This makes sense. So if I compared my and my sister's DNA, we would be really similar, more similar than me and my cousin's DNA. And even more similar than me and some random person I've never met's DNA. And we can kind of expand that even outside of humanity. Okay, so humans and chimpanzees, we're pretty similar. Our body plan's generally the same. We're both primates. We both have opposable thumbs. But a chimpanzee is clearly not a human, right? But our DNA is still 96% identical to a chimpanzee. So really, really similar because, again, we have really, really a lot of traits in common, and that DNA must be in the 96% that makes us the same. Okay, so humans and dogs. We're pretty different than dogs, right? They have paws instead of hands and feet. They have this big, long snout and things. So if you think, okay, how much of my DNA do I think is the same as a dog? Well, it might still be higher than you think. Okay, 82% identical to a dog. Remember, though, still made of cells, still have mostly the same organs, hearts and lungs and all of those things, and blood vessels. So we need mostly the same instructions. And what about bananas? So now we're pretty different, right? So we're still made of cells, true, but a banana tree doesn't have a heart, it doesn't have a brain, it doesn't have a lot of these things that are really, really what makes us similar to like dogs and chimpanzees. So if you think, okay, how much DNA do I think I have in common with a banana tree? Well, it's still 60%, because you're still made of cells, still have to be able to do those really, really important things here. Let's see. I think I'm going to do one more page here. So structure. So how do we build these things? Okay, so we looked at the nucleotides, but we haven't really seen them put together. So both our nucleic acids, DNA and RNA, have what we call a sugar, I don't know why that U looks so weird, sugar phosphate backbone. Okay, so that means the backbone of DNA and RNA is made of sugars and phosphates, and they just alternate. They go sugar, phosphate, sugar, phosphate, sugar, phosphate. And the sugar of one nucleotide attaches to the phosphate of the next, so we get that alternation. And then the bases are not attached to another nucleotide along the spine. They kind of stick out towards the side. Okay, so again, so here's a sugar, there's a phosphate. So here's the backbone, and it goes sugar, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. So it just alternates. On the other side, same idea, sugar, phosphate, sugar, phosphate, sugar, phosphate. And so they're held together to form the backbone. And they are held together with a strong covalent bonds. Okay, these bonds, if all goes correctly, do not really break. Okay, so I could pull the DNA apart, this strand, pull apart from this strand, but that's breaking these apart, okay, in the middle, not breaking these bonds that hold the backbone together. The backbone hopefully stays together all the time. In DNA, the double strand allows bases to reach out and attach to the base of the opposite strand. Okay, so the base of this nucleotide is going to reach out and be attracted here to this base of this nucleotide. Okay, and it'll kind of, once it forms like this, then it twists around and we get this guy, which we call a double helix. Whoops, what am I doing? Double helix. Okay, and the two strands of DNA in the middle here, where the nitrogenous bases are kind of paired up, and remember here we have apples in the tree, cars in the garage, A's with T's and C's with G's, are held together with what are very, very weak hydrogen bonds. Okay, so if this DNA, if we wanted to, we could separate, we could break this right here, break those hydrogen bonds pretty easily. And then we have one strand of DNA over here and one strand of DNA over here. And we have to do that both when we need to make a copy of DNA, like we talked about, we have to make a copy of DNA during interphase, or if we actually want to read the DNA, we want to read the instructions, because the instructions are here in the order that the bases are in. If we want to read the instructions, we have to open up the book, right? It's kind of what that's happening there. So there is, I know there's one little page left of this section. I'm going to save it. So there's most of 11.1. I'll talk to you later.